Thalassemia is an inherited hematological disorder caused by defects in the synthesis of one or more of the hemoglobin genes. The alpha and beta forms of the disorder are characterized by a reduction or an absence in the production of alpha and beta globin chains. Imbalances in the alpha and beta globin chains cause both a hindrance in the production of new blood cells as well as the breakdown of existing blood cells. There are also a variety of different forms of alpha and beta thalassemia, including silent, minor, intermediate, and major, on an increasing scale of severity of anemia with microcytosis and hypochromia, as well as growth retardation. Mitosis is when your mean corpuscular volume is less than 80 micrometers cubed, while hyperchromia is the red blood cell appearing to have less color than normal when examined under the microscope. Within thalassemia intermediate, you can sometimes have an increase in production of alpha globin chains, but not fully understood why. The production of alpha globin is controlled by two genes located on each of the 16th chromosomes. The deficient production of alpha globin is generally caused by a deletion of at least one of these genes. A single gene deletion will result in a silent carrier, which will be an asymptomatic individual. A two-gene deletion causes microcytosis without anemia and is classified as an alpha thalassemia trait. A three-gene deletion causes production of hemoglobin H, also called HbH. This is called HbH disease. The intermediate form of alpha thalassemia results in microcytic anemia, hemolysis, and splenomegaly. A 4 gene deletion results in alpha thalassemia major with HB Bart's, which is typically fatal to fetuses. While mutations in the HBB gene are located on chromosome 11, position 15.5, are related to beta thalassemia. There have been more than 200 different mutations cataloged for beta thalassemia. Most are point mutations along with some rare deletions. The HbA1, HbA2, and HbB genes are responsible for the production of alpha globin and 2 beta globin chains. It takes 2 alpha and 2 beta globin chains to make adult hemoglobin, HbA, and the same for HbA2, but with a different beta globin chain. Hemoglobin is responsible primarily for carrying oxygen throughout the body. Hemoglobin is also an essential storage complex for iron within the body.
One study found a total of 20 different mutations in the coding region of the HBB gene. The most common type of mutation was a missense mutation, accounting for 83.2% of all mutation types. Missense mutations result in a different amino acid being added to the protein product. This can cause a global chain to lose its affinity to bind to its matching globin chains, hindering the production of hemoglobin. There is also an occurrence of mutations occurring in the splicing region. The mutation IVS2-654 creates aberrant slicing, leading to the retention of nucleotides 580-652 of the second intron. The addition of 24 additional amino acids causes the globin chain to lose its binding affinity, while deletions completely remove the ability to produce either alpha globin or beta globin. The exact mechanism is not fully known, as erythropoiesis is a complex process that involves a balance of proliferation, maturation, and apoptosis of red blood cells. There has been a study that shines some light with one mechanism responsible for beta thalassemia major. It says that alpha globin chains perform sequestration of the HSP70 chaperone. This leaves the nuclear GATA1 factor unprotected, leading to the cleavage at the beginning of terminal differentiation, stopping both apoptosis and differentiation. Looking beyond the limited knowledge of the exact mechanism, the issues with the correct amino acid sequence, and other factors that help control erythropoiesis, we are able to understand how these issues lead to other problems within the body. For this, let's focus on just beta thalassemia because of the excess free alpha globin chains clumping into inclusion within the cell, causing hemolysis, red blood cell breakdown in the bone marrow. Hemolysis causes red blood cells to spill into the bloodstream and is broken down into bilirubin and iron. After some time, the buildup of bilirubin leads to jaundice, and the excess iron causes secondary hemochromatosis, which is excess iron absorption. There are also macrophages in the spleen that will destroy the red blood cell in a process called extravascular hemolysis. Hemolysis leads to hypoxia, low oxygen levels in the blood due to a lack of red blood cells. This in turn signals for the production of red blood cells, which can cause the skull, liver, and spleen to increase in size. It is expected that someone in the beta thalassemia major will not show symptoms until they are 3 to 6 months old due to the use of fetal hemoglobin that uses no beta globin chains. You will expect to find anemia and growth retardation from the lack of red blood cells.